I'm Guozhen Liu from the School of Medicine, the Chinese University of Hong Kong at Shenzhen. So today, my talk's title is CRISPR-Cas biosensing systems. We are talking about the opportunities and challenges regarding these CRISPR-Cas biosensing systems. So, my talk's outline includes the introduction of the CRISPR-Cas systems, advantages of the CRISPR-Cas biosensing system, and the challenges of CRISPR-Cas biosensing system. And also, finally, we will talk about the CRISPR-Cas systems towards next generation of the biosensing. As we know, the CRISPR-Cas system begins at genomics, genomic editing, as we know, and also they got the Nobel Prize in 2022 regarding the, the Nobel Prize in chemistry is 2020. So regarding the CRISPR-Cas systems. So since they got the excellent performance in genomic editing, they also got the very proud, successful journey in the diagnosis since 2017. So another pioneer researchers in CRISPR-Cas system, and besides Emilia Chapman-Turner and Jennifer Doudna and Professor Feng Zhang, also the pioneer of this CRISPR-Cas system, especially for diagnostics. So what is CRISPR? CRISPR means clustered, regulated, interspaced, shorter palindromic repeats. Cas means is CRISPR-Cas associated, and cRNA means CRISPR RNA. So that is what is name for that. So, so this is a basic introduction of CRISPR-Cas system. Before we go to talk about how we can apply the CRISPR-Cas system on the biosensing, we have a look, what is a biosensor? How does a biosensor work? We know a biosensor is a medical device which can provide the information of the target analyzed in a solution or in a system. So we wanted to tell about this biosensor is going to tell what if this your target analyte is there, or how much of the target analyte is there. So for biosensors working principles, we start from samples. That means when you want to develop a biosensor, you first need to know what are samples of your analytes in. For example, in the blood sample, we have the food sample, or we have the urine sample, etc. After that, you need to be very clear about your analytes. What is the nature of the analytes? For example, is it small molecules, is it big molecules, the proteins, or virus, etc. So if we wanted to develop a successful biosensor, we need to be very clear about our target analytes and in what samples we needed to detect. After that, we needed to deliver a sensing system which can do the sensing work. We need to have the biorecognition molecules, that means it can specifically recognize our target analytes. After that, we need to have tools for the signal transducers. That means what kind of transducer you're going to choose. That is like chemical transducers, optical transducers, or thermometric transducers, etc. Finally, we need to have the signal processing and state. So we can see here, how does a biosensor work? That at least we needed to have three compulsory components in a biosensor. That is, we need to have biorecognition molecules, signal transducer, and signal processing, right? As long as we have that one ready, then we can have a device can do the work for the biosensing. So this is is to explain how does a biosensor work. After that, then we develop a biosensor, we need to categorize a biosensor's performance. So that includes sensitivity, stability, response time, specificity, and selectivity, and linearities. So today, our topic is CRISPR-Cas biosensing. That means how we develop a CRISPR-Cas biosensors for the sensing performance, and have these characteristics, how we can make sure these characteristics, performance of biosensors is desirable. So we have a look how many types of this CRISPR-Cas system can be used for biosensing. That is from genomic editing to biosensing. So far, we have lots of these several Cas proteins can be used for the biosensing. For example, that is Cas9 and Cas12a, Cas13a, and Cas14a, and even more. This kind of Cas system is developed with the development of the biotechnologies. So as we know, we have different types of these types of the proteins, right? We have these type 2, type 4, and type 5, etc. So this we can see here. So effective proteins have different ones. They have PAM sense the sequence are differently in different types, and also have target. For example, if we use Cas12a, our target it should be the double-strand DNA or single-strand DNA. And for then also they have the endonuclease. This is what it looks like. And also if they can do transcleavage, so different. So cis cleavage is recognized and cleaving the target sequence, make CRISPR-Cas highly specific for biosensing. For the transcleavage is degradation of single-strand DNA or single-strand RNA reporter, make CRISPR-Cas outstanding sensitivities for biosensing. So this is two characteristics related to CRISPR-Cas biosensing system, that is the cis cleavage and the transcleavage. So now let's have a look of the history of the CRISPR-Cas biosensing. So the timeline for the important roadmarks for the development of CRISPR-Cas based technology actually is started in 1982. That is CRISPR named, right? But at that time, they not been used for the biosensing yet. The actual biosensing started from this about 2016 or 2017. You know, in that time, they have the Sherlock, the very famous Sherlock CRISPR-Cas biosensing system that is used Cas13a as these proteins to do the biosensing. And several others, for example, Cas9 and based this biosensing system. And with this time going, lots of more and more powerful CRISPR-Cas biosensing systems are developed. And even now, they have the commercialization for this CRISPR-Cas based diagnostics. For example, these industries in CRISPR-Cas based diagnostics, the Sherlock diagnosis, Proof diagnosis, Turloby. So all these kind of industries are showing up. That is because of the success of the CRISPR-Cas system in biosensing and all in diagnosis. So that is very good. The good science for our CRISPR-Cas. That's all why so many researchers also working on CRISPR-Cas biosensing, because that is a very powerful techniques for use biosensing. As we know, the CRISPR-Cas system, this starts from the genomic editing, but now we use for the biosensing. However, this target analyze not just the nucleic acid. They can far beyond the nucleic acid. For example, we can use this CRISPR-Cas system for use for detection of the proteins and small molecules, and also can use detection of the metal ions, and even for detection of the anesthetic transcript factors, etc. So that means they have lots of various targets can be used, can be applied for by this CRISPR-Cas system to detections. Alternatively, that means we can use CRISPR-Cas biosensing system for detection of the various of the target analyzed, that is beyond the nucleic acid. So how we can apply this CRISPR-Cas biosensing system for proteins? So the things we can do is a very popular way we can do is convert protein signals to nucleic acid signal. We can label this single-strand DNA on the antibody, the part here. Then as soon as we develop the assays for proteins, that means we form the sandwich assay. We have this immunoassays for the proteins. As long as we have the antibody there, we can label this antibody with the single-strand DNA. And as long as this single-strand DNA can trigger the CRISPR-Cas, that means the CRISPR-Cas can be applied successfully for this detection of the proteins. For example, previously we have successfully applied, modified a single-strand DNA to detection antibody of this insulin, right? And then this one have successfully used for detection of insulin with the sensitivity of the 10 picogram per mil, so very high sensitivity. So antibody conjugated with trigger single-strand DNA by strep-evident binding to applying CRISPR-Cas platform for protein detection. So this means in order to attach this single-strand DNA to antibodies, there are several strategies. For example, we can use this strep-evident and biotin linking. We also can use the clicker chemistry. We also can use the EDC and HS coupling to realize the single-strand DNA labeled with antibodies. Beyond that, we also can targeting the single-strand DNA auto antibody and also help to increase the sensitivity of the ELISA. So we know we're using ELISA. We have the sandwich assay, right? In the bottom part, we have the capture antibody, then we have the target analyze, then we have a detection antibody. And then this detection antibody will labeled when a single-strand DNA, they have the signals will be amplified significantly, even can increase for 10⁻³ orders higher. So that means we can significantly increase the sensitivity of the ELISA after applying this CRISPR-Cas sensing system for detection of the proteins. So that is also the beauty of the CRISPR-Cas. So we not just can use CRISPR-Cas biosensing for nucleic acid, we also can be used for detection of the proteins, small molecules, etc. And also alternative, and another attractive point of the CRISPR-Cas is for the signal readouts. So signal readout, the most popular things we use for readout the fluorescence. However, with the development of this technology, we not just use the fluorescence, we also can use for the colorimetric and also even for the chemistry with the signal readout. Okay, so that is another attractive point for the CRISPR-Cas biosensing. So as we just mentioned, we start this CRISPR-Cas system from the genomic editing. Now we apply for the biosensing. For the biosensing, we know we needed recognition molecules, we needed transducers, and we needed the signal processing. So just now we already just like to summarize or some discussed regarding the CRISPR-Cas system, we can use it as is using nucleic acid as a recognition molecule to trigger the CRISPR under support, not just using this system for detection nucleic acid, because we can label this single-strand DNA that is a trigger DNA to the antibodies, can realize the proteins detection and also other molecules detection. For the signal readout, so the popular thing is you have the fluorescence, but we also can use other signal readout strategies. So that is the attractive point of the CRISPR-Cas. There are lots of varieties we can select it. And also beyond that, we can also have the very successful applications for the CRISPR-Cas sensing in the lateral flow assay. We know the lateral flow assay is a very popular point of care detection system. For example, we use it for the HCG, that is pregnancy test strip, and also even for the last 3 years of the COVID-19, we can develop this test strip for detection of the virus infections. So we also can apply the CRISPR-Cas here to realize very sensitive colorimetric detections based on the lateral flow assay. And also the people realized these detections not just using the bulky machine and like ELISA fluorescence reader, and also we can use these optical signals based on the mobile phone using the realize the digital signal readout. So this is how to integrate the CRISPR-Cas system to the point of care detection and also integrated with the digital equipment such as the mobile phones can realize this smart or intelligent diagnosis based on the CRISPR-Cas systems. So here is also another example using the color or the photothermal changes based on CRISPR-Cas. So in this example, we can see here our target is a bacteria, and the bacteria, and we extract the DNA, and then using we got this double-strand DNA, and then using it as a trigger analyze, right? So we can trigger this Cas12a, and then they can realize the fluorescence. So this is fluorescence detections, fluorescence-based detection based on using our CRISPR-Cas. And also here, based on the aggregation of the golden nanoparticles labeled with DNA sequence, we can see the colorimetric change. For example, before and after the analyze, from this here, the color can change from the purple to red. And also this can realize the heating change as well. So because this change can generate the heater, that means you can realize the dual mode detection based on CRISPR-Cas. That is colorimetric-based detection and also photothermal-based detection. So, and also this whole process, you can see here, is DNA extraction and PCR amplification about 50 minutes. And then we can have this CRISPR-Cas12a, Cas12 cleavage of the link single-strand DNA 5 minutes. And then we can realize 5 minutes colorimetric-based detection and 5 minutes photothermal-based detection. So we can see here the CRISPR-Cas, the powerful point is it can be integrated different system to realize various detection platforms here. And now I want to show one example showing how to integrate of the CRISPR-Cas sensors with the surface enhanced Raman spectra and also with the micropath detections. So this one is using the detection of the food quality or food safety. So firstly, we have some food, and we extract our target double-strand DNA from our the food, right? And then we develop of the CRISPR-Cas12a system to generate the fluorescence signals here. So this is, we not realize the fluorescence signal in this system because we wanted to apply the source, the detection of the optical sensing readout with our paper device. So in this one, we applied the golden nano stars. So this golden nano stars, right? Then we labeled with this, we linked with the single-strand DNA, right? And based on the single-strand DNA, when we did do hybridization, they will have this together, have this linked, linked with single-strand DNA. And without the target analyze, they just generated like that. However, with the target analyze, this is triggered, and this will be coded by our CRISPR-Cas systems. And then these golden nano stars will be separated, and then they have generated the colors or this kind of source tag. These signals will be picked up our source, the handable and the portable, this source instrument. So you can see here. So with the presence of target analyze, and this we got the source tag released, right? So before, without the target analyze, no source signals, and after they analyze with higher source. And also we apply the micropath. What is a micropath? Is a microfluidic paper-based analytical device. So on the micropath, you can see here, if they have generated the signals, we drop there. And then because this is, with the presence of the target analyze, this golden nano stars, they will move along this paper device, this microfluidic paper device. They will from left to right. And then we apply the source, then we can pick up this source signal of the target analyze. So this method is very sensitive, can reach 3 to 4 CFU per mil for spec'd milk and meat samples with a dynamic detection range from 1 to 108 CFU per mil. So this is based on the RPA Cas12a and micropath based the detection, and can successfully apply for the food safety and quality detections in 45 minutes. So actually this paper is published in Biosensing Bioelectronics last year and got a very high citation so far. So if you're interested, you can read these papers online. And now we also give another example showing the potentials of the CRISPR-Cas biosensing, that is digital microdroplet-based CRISPR-Cas biosensing systems. So here is to use for this SARS-CoV-2 the RNA detections. So you can see here, so inactivated SARS-CoV here, and then when we using the single-step Cas12a assisted risk transcriptors RPA. So this one, when we have the target analyzed through this RPA, they will generate this, they will trigger our Cas12a to generate the fluorescence signals. You can see here, these flow signals. And these generated signals, we can generate these droplets based on this the microfluidic, right? So then we can have the droplets. Each droplet will fill in the wheels, and then we can realize the digital counting of the wheels of the generated signals, can realize the digital quantification of the target analysis. So this is another beauty of the CRISPR-Cas biosensing system. They can integrate it with these micro droplets to realize digital detections. So so you can see here. So when we so this is another example showing how you can see ultra localized this Cas38 assay. This is the assay here with targeted RNA. So then we have the CRISPR-Cas, the reagent, they mixed together, and then they generated the picometer sized reaction array, and this array, the droplets for here, and then they can fill in these wheels and one by one, and then you can realize this digital countings, because this each one they can generate, even they have the target analyzed, this one be cutted and they will fluorescence off, fluorescence on, and you generated the signal, and then you have this microscope on this top of the chip, droplet chip, and then you can realize the digital quantification of this your target analysis based on CRISPR-Cas biosensing system. So this is these two examples showing the CRISPR-Cas is very adaptable to other systems to realize these very sensitive assays, and also even can be applied, can be useful in the point of care detections. So just now the two examples or several examples have shown the potentials or success of the CRISPR-Cas biosensing in different application scenarios. They can use for the healthcare, also we can use for the food safety, and also we can see this CRISPR-Cas biosensing not just simply to generate a fluorescent signal, they also can generate the other signals, for example, colorimetric, and also optical signals, for example, the search signals, and even the electrochemical signals. So all these potentials confirms or justifies or showing the advantage of the CRISPR-Cas system. They can do the disease diagnosis and management. They can do public health, and also can do the epidemiology in studies, and can do the environmental monitoring, and can do the food safety and quality control, and even can be applied in biotechnology and drug delivery or drug development system for that. So this picture just showing the just to see the wide application or advantages of the CRISPR-Cas biosensing. You can see here. So they can have the CRISPR-Cas, they can realize the point of care detection. Just now we show you in the paper lateral flow assay, right? And also we just show using the micropaths based search detection using the CRISPR-Cas biosensing. And also they can realize the multiplex detection. So in this multiplex detection, you can different strategy we're going to talk about in the following slides. So, and also they can do the real sample analysis. And because if we have this good sample treatment chips or modules integrated sensing device together. And also we have different amplification methods. Just now we see the RPA, and also we can use in the RCA and LAMP, other biotechnologies for the single amplification with this CRISPR-Cas. And also we can do the single nucleotide polymorphism detection, just SMP detections as well. That is further confirming or showing the sensitivity and of the CRISPR-Cas systems. So this is showing the different application methods based on CRISPR-Cas. So now the question is, are CRISPR-Cas biosenses really sensitive? Because we show the just already show the this CRISPR-Cas biosensing is very sensitive and very specific. As we know, almost a majority of the biosensing CRISPR-Cas system, they used CRISPR-Cas, the Cas12a or Cas13a. But they are very difficult to generate collaborative activity when the amount of targeted nucleic acid is less than picomole levels. That means majority of time, CRISPR-Cas biosensing requires pre-amplification steps, that is RPA, RCA, LAMP, this kind of amplification strategies to realize the sensitive detection. So pre-amplification is very popular used in CRISPR-Cas biosensing in order to realize very sensitive detection by combining with other nucleic acid amplification methods. For example, we can integrate it with different nucleic acid amplification techniques. However, we needed to choose which technique is best to use for the pre-amplification, because all these different NAATs, but the requirement are different. For example, the reaction temperature different, the maturity stage are different. But we know CRISPR-Cas sensing, they can realize the sensing system happen at room temperature. So ideally, we need to have something technology also can do the pre-amplification at room temperature. For example, the RPA, RCA, etc. Right? If we use a lab, that means we need a very high temperature, cannot realize the detection at the room temperatures. So you can see here, integration of CRISPR-Cas with the nucleic acid amplification technologies. You can see firstly, we can do the single amplification. That is our target analyze, could be DNA, could be RNA, even also could be the proteins. So we can do the RPA, also we can do the PCR, and also this is a reverse transcript PCR, and also we can do the real-time PCR, and also we can do the LAMP on the real-time NAM etc. here, RT LAMP. So then we can do the signal transducings. We can see Cas12a CRISPR-Cas biosensing, right? And also we can use in the 12a-B etc. So then we have signal reportings. You can for this signal, also can generate the colorimetric signal or either chemical signals. So that is the general principles or scenarios how we can integrate the pre-amplification technologies with CRISPR-Cas biosensing to realize these sensitive detections. So then also in order to further increase the sensitivity of the CRISPR-Cas biosensing, besides using the nucleic acid pre-amplification, we also frequently use nanomaterials combined with the CRISPR-Cas biosensing. So these nanomaterials, we can have a look here. Maybe slow. Yeah, here. For example, this is we apply the gold nanoparticle related to CRISPR-Cas biosensing system. So this is 20 nanometers gold, and this is 60 nanometers. Then we can use a single strand DNA linked, and then we have another single strand DNA to do the hybridizations, but here labeled with this fluorescence, because this when the fluorescence is close to the golden nanoparticles, it can be quenched. So, but if we do the cutting, and then this fluorescence will be shining on, so then we can realize from fluorescence off to fluorescence on. So you can see here. So that is after this CRISPR-Cas reaction happens, they do the transcleavage reaction of a single strand DNA, and then it can release a fluorescence signals, that is from signal off to signal on. So it can give very sensitive assays. So you can see here, for the CRISPR-Cas biosensing, we can apply the nanomaterials into the system to realize this more sensitive biosensing. So another example using the nanomaterials combine the CRISPR-Cas biosensing using the graphene oxidase. So that one is can realize this. So we can, before CRISPR-Cas sensing attach these golden nanoparticles on this graphene oxidase transit system here, and then after that they can after the transcleavage effect of the single strand DNA, they can realize from strong sense intensity to realize weak sense intensity. So this is realize signal off detections based on using applied nanomaterials. That is also help to increase the performance of the CRISPR-Cas biosensing, especially for the sensitivities. So that is all showing the powerful of the CRISPR-Cas biosensing. Now is another important application for the CRISPR-Cas biosensing, or beauty of the CRISPR-Cas biosensing is used for the point of care testing. Just now we also show how we can apply the CRISPR-Cas biosensing in the lateral flow assay to realize the point of care testing. So you can see here, so nucleic acid-based single application, different strategies. We can apply all this together to realize the point of care testing. So this example, lateral flow assay combined CRISPR-Cas system, they are very cheap, rapid, and portable. And we just need to realize yes and no, the diagnosis, very quick. So you can see here, so we can link this CRISPR-Cas, this reagent on the paper substrate in the test line or control line. And as soon as the Also, of course, we need labels. This label that we use called the nanoparticle labeled here. As soon as this target analyze present in the sample, this CRISPR-Cas will be triggered, and then this nanoparticle will be cut it off, and then they can move to our T line on the C line. Then to realize this biosensing is here. So that's a very successful example use the CRISPR-Cas biosensing into paper lateral flow assay. We can see here, we still rely on gold nanoparticles because these gold nanoparticles help us to realize this intensities change in T line or C line. And another example use this CRISPR-Cas biosensing in the point of care testing is integrated microfluidic paper-based analytical device, and also integrated with a paper origami means. So this example is published in Nature Biomedical Engineering in 2019. So basically, the people wearing the mask, and then the developer this device integrated the paper origami into the mask. So when we breathe, there will be aerosol. The aerosol will be trapped in this collection zone. As well as this one here, they have a button where you press it, and this reagent will be flow through this collection zone, and then the target analyzed virus will be reached to the lysis zone. Then the virus will be lysed, and the RNA will be released. This RNA release will trigger the RPA, and then following this Sherlock, that is CRISPR-Cas detections, and then they generated the signal. This signal will be displayed through the paper-based lateral flow assay. So you can see here for the lysis, and then this isosomal application, that is RPA, and then we realize Cas12a, the Sherlock detections, and generated the signals, and also convert that fluorescent signal to the colorimetric signal of the, you can convert the signal to the gold, and also you just directly use this physics. This is only physics conjugated gold nanoparticles, right? And then you can realize these colorimetric signals on the paper lateral flow assay. You can see this device, integrated device, they integrated wearable device. They are non-invasive and also end-user friendly, because we don't need to do any sample treatment, and also is very safe. And the CRISPR-Cas detection and the paper lateral flow, and also microfluidic paper-based analytic device. This is truly point of care testing or point of care diagnosis. So this design is very smart. Actually, the principle is not so difficult. You just collect the sample, and then you have this CRISPR-Cas biosensing happening in the microfluidic paper-based analytic device, and then we realize signal readout through the paper lateral flow assay. So that is basically the principle here. So now, in order to have a very successful CRISPR-Cas sensing system towards next generation biosensing, what are the shopping list? What in this wish list? That is, we need to make sure they are very high specific and high sensitive, and also need to be very rapid detection, and you can realize in the point of care testing. And also, hopefully, we needed to integrate abundant target amplification strategies, and also need to have various signal transition enhancement and output methods. And ideally, we need to have multiplex detection, and also needed to integrate plenty of the automatic, integrated, portable, or cheap analytical platform. So this is our wish list. When we design a CRISPR-Cas biosensing system, we need to think about it. How many kind of items we can consider can be integrated together to realize the best performance of the actual applications. So we can see here the real application we realize is a portable all-in-one analytical device. That is a portable device, and then quickly feedback to users, the patients digitally. And we just needed to have this reaction happen. For example, this signal can be reported to your mobile phone, and also other tools can eventually your personal equipment can collect lots of images, and then you can realize the artificial intelligence or comprehensive assessment based on the detections. This is ideal scenarios, how we can make sure this diagnosis happen in this end-user friendly way. That is the next generation of the biosensing. So how we can use this CRISPR-Cas sensing system here, then we need to make sure in this all-in-one analytical device, how we can implement this CRISPR-Cas biosensing in our real target applications. So that's this is CRISPR-Cas system towards next generation biosensing, and the perspectives is sample treatment and one-pot reaction. So we need, without any sample treatment, we can do one-pot reaction. We realize sample in and answer out, and also can significantly increase the signal noise ratio, and also amplification-free method. We don't need any pre-amplification, because although this pre-amplification, they are very sensitive, can increase the performance of CRISPR-Cas biosensing, but this type of the pre-amplifications, they increase the detection time and also increase the cost. Most importantly, they also sometimes introduce these false positive signals. And also we don't need the cold chain transportation, and also realize multiplex detection. So in the future, for the CRISPR-Cas biosensing, we need, from these 5 aspects, consider how we can increase the system to realize this wide application of the CRISPR-Cas biosensing from humans to animals to plants to bacteria to virus to exomes, and from nucleic acid to proteins to metabolites, from single molecule detection to multiplex detection, from in vitro to in vivo molecule monitoring. So that is the future of the CRISPR-Cas biosensing, our wish to apply various wide applications for that. Here, I also would like to acknowledge my team. So my team is ID2 and also the funding agencies to help to support our research. So finally, thank you all for your attention. That's all for my talk. Thanks.